I'm Dr. Jim Zoval. You're about to watch one of my video lectures. If you like this video and would like to get videos for your entire general organic and biochemistry course, you can do so at www.zovallearning.com. I'll give you more information about that at the end of this video. Let's recall our definition of a stereoisomer. Molecules that have the same formula, the same molecular formula, the same atomic connections, but different three-dimensional shapes. We saw a particular type of stereoisomer in chapter 4 called a geometric isomer. Geometric isomers come about because of restricted rotation. Sometimes when you have a double bond, you could have a geometric isomer because double bonds can't rotate. We also saw geometric isomers in cycloalkanes. When we have a couple substituents in a cycloalkane, we saw that that could be cis and trans also. For the same reason, those substituents can't rotate through the center of the ring. Restricted rotation causes geometric isomers to occur. Another type of stereoisomer occurs when 4 different groups are bonded to the same atom. These videos are made to be viewed while using fill-in lecture notes. The pause in the audio is to give students time to complete the lecture notes fill-in. The lecture notes can be downloaded, free of charge, at www.zovallearning.com. In these cases, there are 2 distinct three-dimensional arrangements of the atoms. For our purposes, we'll only be concerned with the pairs of stereoisomers that result whenever 4 different groups are bonded to the same carbon atom. A carbon atom that carries 4 different groups is called a chiral carbon. Bear with me for a moment. I'll give you quite a bit of terminology in this slide, but there'll be plenty of visualizations to go along with it shortly afterwards. The 2 distinct, three-dimensional arrangements of the atoms around the chiral carbon are mirror images of each other. When 4 different groups are arranged in the tetrahedral geometry, we called it A before in a previous chapter, the mirror images are not identical. Another way to describe non-identical mirror images is with the term non-superimposable mirror images. Stereoisomers such as these, which are non-superimposable mirror images of each other, are called enantiomers. Thank you for your patience. We made it through the terminology of this slide. I don't expect that to make a whole lot of sense to you yet. I wanted you to have it in your notes, but now I'll elaborate on it and give you some visualizations. Enantiomers are non-superimposable mirror image molecules. Now on 3, you guys will say, huh? 1, 2, 3. Which is appropriate. There's a lot of stuff in that sense. Let's unpack it. Let me give you an example you're familiar with of a non-superimposable mirror image. Your left hand looks in the mirror and sees what? Your right hand, right? So these are mirror images. Sometimes mirror images can be superimposable, which means you put them in the same place in space and they look identical. Let's try that with our hands. So we'll put them in the same place in space. Let's say I was able to get the... You see, it's not quite identical because one hand is facing up and the other hand is facing down. So if I turn my hand over to make them, you see how it's not identical? The thumbs don't line up, the pointer finger and the pinky finger are there. So these are non-superimposable mirror images. An example of a superimposable mirror image would be, say, a sphere. Let's say you have just a sphere, right? It looks in the mirror, it sees a sphere. You could put those in the same place and they're identical. In the case of molecules, these would be 2 different molecules. These would be enantiomers. In the case of the spheres, it would just be the same molecule. They're not 2 different ones. Let me show you another example of a molecule that has a mirror image that is superimposable. Notice the black sphere, that should represent carbon. We're always going to look at carbons because we're doing organic chemistry. I have a red sphere, a yellow sphere, and a green sphere on these. Those would just represent different groups. Could be methyl, ethyl, hydrogen. It looks like they're looking in the mirror, right? This one looks in the mirror and sees this one, right? Notice they're superimposable. I'll bring them together and everything lines up. Right? The red lines up with the red, the yellow with the yellow. So these 2 mirror images, they're identical. That's the same thing. Okay? When you have a carbon with 1 or 2 or 3 groups on it, their mirror images are superimposable. It's the same molecule. There's no difference. However, let's look at what happens when we have 4 groups attached to a carbon. So here, this is supposed to represent a model of a molecule, carbon in the middle. I'm holding the carbons with 4 different groups attached. Orange, yellow, green, purple. Okay? This could be hydrogen, methyl, ethyl, propyl, just 4 different groups, right? So this one looks in the mirror and sees this, right? Green, yellow, orange, then back behind the purple. Right? Let's try to superimpose these. So let me just line up 2 of the colors. I'll try to line up the orange and the purple. And it looks like it's been superimposed, but if we look at the other side, look at the green and the yellow are off. Okay? If I line up the green and the yellow, guess what's going to happen to the orange and the purple? Right? Non-superimposable mirror images. These would be 2 different molecules. Their properties would be a bit different. To get this to be identical, what I have to do is I have to change 2 of these. Now they're identical. They could be superimposed. Green, yellow, everything lines up, but they're no longer mirror images. It's the same molecule. Enantiomers are non-superimposable mirror images. So now when I read this sentence, enantiomers are non-superimposable mirror image molecules, you guys don't say, huh, you say, oh yeah. All right. I have a question here.
Why is this important?
Very good question. And then you guys should always ask that. We're going to see why this is important. Let me give you a little insight to why it's important. When you read your biology books and you read about a molecule that's called glucose, right? Why is glucose kind of important in biology? We get energy out of it, right? And how does, what makes the glucose? Where does it come from? Magic? Yeah, almost. No, but where do we get it? Where does food get it? You guys know the answer to this. I'm calling Huntley and Tay over here. You guys? Thank you. Photosynthesis, right? CO2 and H2O plants do photosynthesis. They do chemical reactions and make it into glucose. Now, glucose comes in 2 forms. They come at 2 mirror images. One's called D-glucose and one's called L-glucose. Right? You guys may have remember reading about glucose and it says D in front of it. That's a particular stereoisomer. Nature happens to make D. We're not sure why that is. It could have been a flipped coin. It wouldn't have been efficient for nature to make both. Enzymes are involved in those reactions, and enzymes are stereospecific. They only make one particular isomer. They only make the D isomer. Guess which isomer we're able to metabolize in glycolysis? D-glucose, yes.
You're looking at an example of a pair of stereoisomers, an enantiomer pair, that results from the presence of a chiral carbon. The carbon is chiral because it's bonded to 4 different groups: a hydroxyl group, OH, a chlorine atom, a hydrogen atom, and a fluorine atom. Recall that in wedge and dash illustrations of 3-dimensional objects, solid wedges indicate bonds that would be coming out and above the page, towards the viewer. Dash shapes indicate bonds that would be coming out and behind the page, away from the viewer. Regular lines, neither wedge nor dash, indicate bonds that would exist on the plane of the page. This image may help you to visualize and understand that the 2 molecules shown in the previous figure are not identical. I've graphically depicted the failed attempt to superimpose the pair of enantiomers. Note that we cannot superimpose these molecules. The groups don't line up. If we try it this way, the chlorine and the hydroxyl groups are not lined up. Stereoisomers such as these, which are non-superimposable mirror images of each other, are called enantiomers. Pairs of enantiomers have very similar physical properties. For this reason, they're very difficult to separate from each other. For example, their boiling points are so similar that separation by distillation is not possible. They do differ in a couple of important ways. Since enantiomers' arrangements of electrons are mirror images of each other, they interact with light in different ways. It's for this reason that enantiomers are sometimes called optical isomers. Another important difference in enantiomers is the way they behave in biological systems. Since enantiomers don't have identical three-dimensional shapes, they don't behave identically when interacting with biomolecules such as enzymes or the receptors that are responsible for taste. The pair of enantiomers of the amino acid named asparagine are shown in this image. One of the enantiomers, called L-asparagine, has a sweet taste. The other enantiomer, Diastereomers, has a bitter taste. L-asparagine is produced in high concentration in the asparagus plant where it was first discovered and got its name. You'll learn the details of the L and D designations for enantiomers in later chapters. Many enzymes are stereospecific. This means that they'll catalyze a reaction for only one particular enantiomeric reactant and/or will catalyze the formation of only one enantiomeric product. For example, the antibiotic that we call penicillin is made by enzymatic reactions by mold. Only one of the enantiomers, penicillin, is produced. Plants produce only one enantiomer of the glucose molecule, D-glucose, as I mentioned earlier. It's therefore understandable that all organisms, with only one known exception of a particular bacterium, will only metabolize the D-glucose enantiomer. The biological production and metabolism of glucose co-evolved, so it makes sense that nature selected the same enantiomer upon which to rely. Let's do an example problem in order to help you identify chiral carbons. The line bond structure of 2-bromo-3-chlorobutane is shown here. How many chiral carbons are in this molecule? To solve this, we'll consider each carbon individually. A carbon is chiral if it's bound to 4 different groups. Carbon number 1 is not chiral. It's bonded to 4 different groups, however the 4 groups are not all different from each other. 3 of the groups are hydrogens. I've highlighted the 4 groups that are bonded to carbon number 1 in this structure. Carbon number 2 is chiral. It's bonded to 4 different groups. A mistake that chemistry students sometimes make is to consider only the 4 atoms to which the carbon is bonded. If you were to do that with this molecule, you might think that carbon number 2 is not chiral since it's bonded to 2 other carbons, carbon number 1 and carbon number 3. You must consider the entire group of atoms to which a carbon is bonded in order to determine if that carbon's chiral. It's for this reason that I've highlighted the entire groups that are bonded to carbon number 2 in the structure here. I'll place an asterisk next to the chiral carbons in this molecule. So far we have 1, carbon 2. Carbon number 3 is chiral. It's bonded to 4 different groups. I've highlighted the 4 different groups. Carbon number 4 is not chiral. 3 of the groups are the same. There's 3 hydrogens. So there are 2 chiral carbons in 2-bromo-3-chlorobutane, carbons number 2 and 3. Before I have you do a problem, there's one last concept I'd like to introduce you to. When more than one chiral carbon is present in a molecule, then more than one pair of enantiomers will exist. The number of stereoisomers that can exist depends on the number of chiral carbons. If we let n represent the number of chiral carbons in a molecule, then the maximum number of stereoisomers is calculated as follows. The maximum number of stereoisomers is equal to 2 raised to the nth power. For example, in our previous example problem, we determined that 2-bromo-3-chlorobutane had 2 chiral carbons. Therefore, the maximum number of stereoisomers that 2-bromo-3-chlorobutane has is 2 to the n, which equals 2 to the second power, which equals 4. There are 4 stereoisomers of 2-bromo-3-chlorobutane. The 4 stereoisomers of 2-bromo-3-chlorobutane, 2 pairs of mirror images, are shown on the right. You learned about the relationship between non-superimposable mirror image molecules called enantiomers. Non-superimposable molecules that are not mirror images of each other but are in the group of 2 to the n stereoisomers are called diastereomers. The enantiomer relationships are indicated with blue arrows. The diastereomer relationships are indicated with yellow arrows. It may be helpful for you to understand the relationship between enantiomers and diastereomers by using a family relationship analogy of siblings for enantiomers and cousins for diastereomers. You'll see more enantiomers, diastereomers, and geometric isomers throughout the biochemistry in this course. Now you try a problem. What's the maximum number of stereoisomers that can exist for each of the molecules shown here? Click pause and work on the problem now. Check your answers. We'll start with the molecule on top. The leftmost carbon is not chiral, it's bound to 3 hydrogens. Moving from left to right, the next carbon is chiral. It's bound to 4 different groups. We'll place an asterisk next to it to indicate that it's chiral. Moving to the right, to the center carbon, this carbon is not chiral, it's bound to 2 hydrogen atoms. Moving to the right, the next carbon is not chiral, it's bound to 2 methyl groups. The rightmost carbon is not chiral, it's bound to 3 hydrogens. So the top molecule has 1 chiral carbon. In our equation for the maximum number of stereoisomers, n is equal to the number of chiral carbons. For this molecule it's 1, so the maximum number of stereoisomers equals 2 to the n, which equals 2 to the first, which equals 2. Now let's consider the second molecule. The leftmost carbon is not chiral, it's bound to 3 hydrogens. The carbon that's second from the left is chiral. It's bound to 4 different groups. The third carbon from the right is also chiral. It's bound to 4 different groups. The rightmost carbon is not chiral. It's bound to 3 hydrogens. This molecule has 2 chiral carbons, therefore the maximum number of stereoisomers that can exist equals 2 to the n, which equals 2 to the second, which equals 4. This molecule will have 2 pairs of mirror images, 4 stereoisomers. You can get your entire general, organic, and biochemistry course as a series of video lectures. The rental period is for 1 year, so you'll have these video lectures for the entire term of the course. You could watch the videos whenever and however many times you wish. I survey my students and they prefer these video lectures over the textbook by a ratio of about 10 to 1. Fill-in lecture notes and an electronic version of my textbook are available free of charge at the website. To get more information or to get the videos, free textbook, and free lecture notes, visit my website at www.zovallearning.com.